Today I will not be in my usual way because I'm little sick and my voice, normally I don't use microphone in this hall, but today I'll be using. So I tried to look around several times from morning, and then I really couldn't find many people who don't know how we are collaborating. And I think I have been collaborating with most of you, even if I count on terms of analysis, talking, designing. I think most of you I have done. So I think one or two hands may be here, which, but still, like I would like to take this opportunity that what I do or what I will do with you, and how we can go ahead. So with me, well, we all are my team members, all readers. So, but still, like those who really help me in day-to-day analytics. As of now, Roma and Anil, I have two special project scientists who work with me. They are present here, and we have some scientific officers who also work with me. So the placement, because this was the first structure, like what our Honorable Director General has sent. Long back, like Statistics, Bioinformatics and Data Management Unit, with reports to DDG with because of its cross-cutting nature, like to all of the Asian regional program and also to both of the global program. So this is the way was the structure. It's also said that both global program also cut across the regional programs, but since this activity cuts across even to global programs, probably that's why it was kept. However, then it was proposed in the last mail what came from DDG from RC members to RC members. I think that might have been forwarded. It was identified that bioinformatics theme fits if in terms of work planning and working like with you, fits quite well even more into the genetic gain. So as of now, in that recent little change in the structure, like bioinformatics will sit with here for research planning and other purposes and working with you. Well, we were working from there, but this is the recent structure, and the statistics and data management still cuts across everywhere. So, okay. This is our operations, SDDM operations. This is a very broad slide. So, and very broad bullet points. If you want, I can go in detail in each of the slides. So, in each of the head. When I say project idea meeting, this is like this kind of meetings where we discuss deliverables of the work, what we are going to do in future, in coming years. So this all operations start like all other disciplines. Start from here, and then here we will those of well, I'm in touch with many of you, but I still like to formalize it better. What kind of things we are? When I say design of experiment, that not per se a field experiment. That's design of your all data related activity in the project. So I could have used a better term, data design, but for better understanding, I wrote because we all do some kind of experiments. So, and then like data collection, when I say field book or lab book, like how, so this is all of the activities which we are doing with you, and we provide support in that, and I look forward to collaborate with all of you in doing all of these things. So we have a full session in afternoon, in evening, so they will talk more. And then data generation protocols, data capture, like in some presentation it was mentioned that we are using barcoding, and then we are supporting with several other things to our readers and other scientists and the other scientists. Data curation is very important, in which we have a very specialized capability, and I think we have given that kind of support to our scientists and also to several CGs and CGR centers. We have been talking, and we have been providing those kind of supports. Even integrated breeding platform people, we have given some several kind of good advisors and ideas on those things. And this is just one part, all kind of analytics using bioinformatics, statistical modeling, survey analysis. I think with last year we got the best paper award with Anthony's group on doing some kind of econometric analysis. So it's quite cross-cutting. So if you want to talk in that session, I can talk more on that. Valid interpretation, publication of result, data archiving and sharing. So I'll talk some other roles and responsibilities to be more clear. Like it's nothing new what I just talked on last slide, but if somebody wants to have clear understanding, here I have written just the tools for the data management and sharing and some so BMS, Dataverse, CMAP, GBrowse, Aware, Green Global, ArcWeb, GIS, etc. These kind of our tools what we are supporting. Some most common frequently offered solutions what we offer to across those boxes in so spatial linkage, QTL mapping, genomic selections, and genome-wide associations, all kind of things. These are the few from our list what we provide. Because if I write all statistical analysis names, so I think we may need 10 slides for that. So I'll highlight this data management support more because in afternoon session we will talk more on that, like what kind of how we are going ahead in this special. So this heart, so heart says that we all have to be in good harmony with each other, and like data team is not different from you. Data team is a cross-cutting with you, and we always support you. So we have to be in good harmony, lovely relationship. So isn't it? Yeah. So I can explain more on that. So well, these are few things in which we are now providing support with our Director General has pushed it very hard. He advised us to implement this modern tool across locations, and we have done that across locations, and we are doing so. Several kind of modern databases we have introduced for our readers. We have done lots of training across several projects. So aim is that data should be available after project life cycle for the next generation, which will when we go from the institute or for better purposes. So then the data should be there, or analytics and those things should stay there in a good and identifiable format. So that's the part. So these are just some digital data collection things what we have talked to our scientists. So when I'm showing this, I mean those who have not worked, but I think everybody has worked. We are ready to provide these kind of solutions to you. We look forward to collaborate. Like we do some dashboarding. I'll show you some dashboards in the afternoon. And there is a very nice initiative, like most of our genomic scientists know, that they are storing their data in some flat file system, and each time when they need to subset it or do something, they need to, I don't know how they do it, because they need to write a script. Many times some bioinformatician help them, or those big 5 gigabyte for one chromosome, those kind of files, they don't have any good system. So in this line we are working in a project. In one presentation you might have seen that. So GoBE is a project in which we are going to establish a high throughput data management system for storing all of the genomic data, sequence data, marker called allelic data that will be sequenced. Like you all, breeders and other scientists, those who wish, they will get their logins there. You can deposit your genomic data there, their sequence are called allelic data. And interestingly, some bioinformatic analysis tools will be there. Like something if you want to subset data on something, like on minor allele frequency, or you want to calculate some linkage disequilibrium. So now you take data from one software to another software. So GoBE will try to overcome these things. So some of the things you can do yourself, and well, we are always there to help you. So if you need some complicated stuff, we are always there. This is planned for locations also. Like we will extend this to location through some kind of VNC like virtualization, or we may have a full-fledged server there also. So of course, and the BMS will be tightly linked with that, so that directly phenotypic data from there directly can go in there. You can run some kind of studies and can do. It's an ambitious project, and we'll update you more on that. This is not very. This is because some people from here, other breeding programs and locations are there, which who are planning to do some kind of farmers participatory trials, farmers variety selection trials. So we have come up with a solution. It's again free, but it has lots of analytical capabilities. It's a multiple language online data collection, simple data collection form with a GPS recorded coordinate. Server is with us. Like it's an ICRISAT server. You can always log in into server. You can see the dashboards. Like it's a simple example. You can take photographs. You can browse it, or you can write something on the fly. It provides several languages. Like you can have several kind of languages here. You can select French, like Telugu we selected for Indian perspective, and then automatically things will change into the language. So that kind of recording. So if the farmers, this can work in simple mobile phone, all Android based or iPhone, all kind of platforms it runs, even on Windows. So it was just an example, like we were in New Delhi, car was moving, and I was recording and taking photographs. So later on you can see that which farmer or which person actually uploaded that point. So you can have a full dashboard. Like if you have 2000 trials, you push this form to all of the farmers, and if they submit the data, you can see it in real time. So this was a photograph, not a field, but something I tried to avoid human, but some guy came immediately. So so like these kind of solutions also we provide you. If you need some of your, they work everywhere where you have a 3G connection. Like you can record data, but once you come to office, then it goes. So offline it works very well. The open access solutions, data management, I invite all of you to share your data sets, what you think are useful for your colleagues sitting in institute, your colleagues sitting outside. So the Dataverse is our prime repository. Again, SBDM is among few 1 or 2 units across CGIR who have the Dataverse 4.2 solution, which is a very vibrant solution. There are several features. So, and this is our own implementation. It's not on Harvard's website, it's on our website. Very fast, blessing, you are fast. So you can see, so here idea is not to just share the files, use something interactive. I'll show you example of some GBS data, which we have kept in there. So you can download the FASTQ file, or even the Cordell file. But apart from that, we are linking these systems. So this system is linked with the virtual visualization flow. Like you just click, if you want to see all of these sequences on GBrowse, you just click, GBrowse window will open from there, and then you can visualize all the features there. And once you close, you are back to the system. Same way, like we got very nice data from Dr. Vani's group. Vani's group. This is a soil sample data here. So here you can see the latitude, longitude wise. There is an Excel file also. You can click and download that Excel file if you want for any purposes. Lots of values are there. All file properties are there. So you can just see. So from this place 215. This is a dynamic map, not an image. You zoom in, you reach to actual location collected. If you zoom out, you can see the interpolated values from those kind of districts. So these maps are online available on this Dataverse website of ICRISAT. We are also collecting weather data, and I invite others, all our locations, if they provide weather data. We have all historical ICRISAT location data here, like you can download it in PDF, table, Excel, pictorial. You can see how the degree is changing from past 30 years. So rainfall, temperature, minimum, maximum, these things are given. You can do lots of analytics. It's pretty dynamic. You don't need anybody's permission to do all this. This is all in open access. Thank you very much.